Hi there, this is David Nguyen, CEO of Quantifies, and in this presentation we're going to talk about how we measured the shape of brain organoids in ways that area could not. We analyzed a paper by Lim et al. From the lab of Jamie Peng at St. Jude Children's Hospital. In particular, we focused on figure S3, in which they showed brain organoids grown to day 55. They compared wild-type cases with 4 lines that knocked out the ATM gene. And in this paper, they measured the size of the organoids and found no difference other than wild type and line 43. But the difference in line 43 are likely due to these very small size organoids, as you can see here. And when you have such difference in size in the same treatment group, there's probably very distinct biology that's going on. So we are particularly interested in figures like this because we can measure shapes in ways that traditional metrics such as area cannot. Because if you zoom in on these organoids, you can see that these lines, especially 3, 14, and 43, exhibit jagged edges compared to the smooth edges of the wild type group. Thus, we felt that this would be a good example on which to prove the efficacy of our shape analysis software known as Genie. So what we did was we manually outlined the shape of the organoids. And the way our software works is that if this is the outline of the object you're interested in, what our software does is it finds the center of that shape, that is the center of the bounding box that encloses that shape, and then it overlays in this case, a radial grid system, which allows us to calculate every intersection between the grid line and the edge of the object. This then allows us to calculate the distance of every single dot to the red center. This then allows us to linearize the data in step 3 into step 4. And in the form of this complex wave, we can apply what's called the fast Fourier transform to turn this shape that we began with into a frequency spectrum. This frequency spectrum is just a multi-dimensional representation of the shape that you began with. And though it is a lot more complicated than what you see in step 1, herein lies a lot of secrets to shape differences that the human eye cannot see. So the way we simplify this frequency plot even further is to derive indices. That is, we can sum all of these bars together to get one number, that is one scalar value. Or in this presentation, what we did was we found certain bars that were different between treatment groups and we created ratios. For example, bin 2 divided by bin 23. That gives us one number to represent the shape that we began with. Now, a very important methodological detail for those who want to replicate this is that for every object, we find the longest internal length, and then we rotate the object such that that length is vertical. And then we find the bounding box and its center to be the center of the radial grid. We do this because nature is inherently polar. That is, North Pole versus South Pole, dorsal versus ventral, so on and so forth. And when we orient objects according to these polarities, we get cleaner data. So let's now talk about what we found. First, we measured these shapes at scale, meaning we measure them as at their original size and shape. And what this does is it compares size and shape at the same time. And when you do that, we find that the data is very noisy, just as the authors had found when they measured area, and that there was no difference in these groups, in these 2 of the 3 indices. And in this third index, derived by dividing bin 3 over bin 24, we did find 2 groups that were different, but not the groups that were visually different, right? As in the groups that we know exhibited very jagged edges. And so this tells us that when we compare size and shape together, it muffles any morphological differences. So what we did next is we measured something called pure shape. As you can see here, we resized each object, right? That is, we normalized it without skewing its original shape. And pure shape is something very special to Genie. As you can see on the left here, these 2 objects are the same shape but different sizes. So if we measure them as is, we measure them at scale. But Genie allows us to objectively resize objects without skewing their shape. In this case, we found the longest internal length, and we resized both objects to have the same height, but we constrained the aspect ratio so that we did not skew the shape. In this way, when we measure shape, we're measuring shape apart from size, which we call pure shape. So this is what it looks like when we measure pure shape. As you can see here, these three objects were resized. Now they all have the same vertical height. And now we see statistical significant differences that match what our eyes visually saw in terms of an increasingly jagged edge, right, as you go from line 2 to line 43 of the knockouts. And here you can see that these two ratios, first one bin 3 over bin 6 and bin 3 over bin 24, do a good job of capturing the difference between the wild type group in blue and the knockout lines 14 and 43, which are yellow and blue. And thus, by measuring pure shape, we find robust differences that are statistically significant. Now, when we put the data of at scale and pure shape side by side, right, it reiterates the point that the differences in shape in these series of organoids is more due to a shape difference as opposed to a size difference. I'd like to contrast the data that you see here with the area measurements in the paper, area measurements that could not see much of a difference between these knockout lines and the wild type group, even though visually we could see and know that knocking out ATM, at least in 3 out of 4 of these lines, results in a much more jagged or folded pattern of the organoids. So what is the significance of folds? Well, brains have folds, and therefore these organoids are embryonic brains, and more folds might mean either more differentiation or uncontrolled differentiation. Indeed, if you look at the data in the paper, they did proteomic analysis of these knockout and found that the proteins that were enriched in these knockout organoids, the theme was neuronal differentiation and maturation, as in proteins that were needed for neurons to become mature and reach out and contact each other, suggesting more differentiation towards the neuronal end of things. And in terms of proteins that were decreased in these knockout lines, the theme was a decrease in epithelial cell proliferation and epithelial cell development. And this makes sense because epithelial cells, a lot of them, such as your skin, come from the same embryonic germ layer as do neurons. That is, they both come from the ectoderm layer. So it makes sense that in these embryonic organoids, as these cells are differentiating towards neurons, they have to turn off the program that makes them epithelial, right? Because epithelial cells, like the cells in your skin, form walls, while neurons were meant to form branches and connections with many of their neighbors. That is the end of this presentation. We hope that you found it useful, and we hope that Shape Genie helps you discover amazing things in your data.